Okay, welcome back. We're fortunate to have Lucy Fang moderate for the next 2 hours. So appreciate many of you who've provided comments in the chat, asking questions, and certainly great opportunity now to do more of that. Much thanks, Lucy, for helping putting today's event together and moderating. So Lucy Fang is Acting Deputy Director and has served as Associate Director of the Division of Quantitative Methods and Modeling in the Office of Research and Standards in OGD. She has expertise in the use of quantitative clinical pharmacology approaches in the review and regulation of generic drugs. She co-leads CDER's work group tasked with the use of partial area under the curve for bioequivalence assessment. Thanks, Lucy.
Thank you, Jim. So hello, good afternoon. Thank you for joining us today. I really hope you enjoyed the wonderful presentation this morning. I know myself, I enjoyed a lot. So now we are moving to the panel session. We have two parts. So for part one, we plan it as responsive to the speakers. I will ask some general questions and as well as some audience questions submitted in the chat box. I will encourage you guys to continue to do that. For part two, we will focus on more in-depth discussion of the model integrated evidence from the best practice perspective. Before we start, I will introduce the 6 panel members who will also join in the discussion in addition to our speakers from this morning. So Dr. Robert Lionberger, is Rob online already? Who also gave us the opening remarks this morning. He is the director of the Office of Research and Standards, OGD. He leads OGD's implementation of the GDUFA science and research commitments, including internal research activities and external research grants and collaborations to ensure the therapeutic equivalence of generic drug products. The next one is Dr. Anna Schwendeman, is the co-director of CRCG William Higuchi Collegiate Professor of Pharmacy and Associate Professor of Pharmaceutical Sciences at the University of Michigan. Her research focus is on the optimization of high-density lipoprotein nanoparticles for treatment of atherosclerosis, sepsis, and drug delivery purpose. The next one is Dr. Bing, who many of you know her. So Dr. Bing is the Associate Director for Scientific Innovation, Office of Bioequivalence, OGD. She provides scientific leadership and expertise for the assessment of bioequivalence studies submitted by pharmaceutical industry through ENDA and oversees the scientific programs, including guidance development and implementation in the Office of Bioequivalence. The next one is Dr. Liang Zhao. Dr. Zhao is the Director of the Division of Quantitative Methods and Modeling, Office of Research and Standards, OGD. Through his 16-year professional career, Dr. Zhao has established his leadership in industrial R&D, quantitative methods and modeling, and model-based strategic decision-making in regulatory and industrial settings for generic and new drugs. The next one is Dr. Raja Velagapudi, who is the Executive Director of Clinical Development at Sandoz. He works in clinical development of small molecules through business development and in licensing for the past 9 years at Sandoz. He has over 20 publications and presented numerous abstracts at scientific meetings. The next one I will introduce is Dr. Tsang, who is Chief Scientific Officer, Biopharmaceutics and Biostatistics at Apotex. To date, he has been involved with design and data analysis of over 1,000 bioequivalence clinical studies for the registration of complex drugs and biosimilar products, and over 300 traditional drugs in Canada, US, EU, and many other international marketplaces. A big welcome to all our panel members. So let's get started with our part one. So the first question I want to direct to Dr. Bing and everyone else as well. So we know that Dr. Ni is from the official bioequivalence. So Dr. Ni, can you provide some perspectives on the current gaps for demonstrating bioequivalence of LAI products from the ENDA review perspective? And also we all kind of discuss on how modeling might help to fill those gaps. So Dr. Ni, I will turn it to you first.
Thank you, Lucy. Can you hear me?
Yes, we can hear you, Dr. Ni.
Okay, first of all, I want to thank all the speakers for delivering such a wonderful presentation. I really learned a lot from this morning's presentation. I can provide some comments from the assessment team's point of view, mainly on what challenge we seeing for the assessment of these LAI products and what we're interested in moving forward. We heard, it is very difficult to design and conduct bioequivalence studies for long-acting injectables. And we heard the current recommendation is a steady state bioequivalence study and very lengthy and requires a long study duration, long half-life, and the drug product is actually designed to have a long Tmax that ended up with, long washout period. And we also heard that, with the study design using patient population, we, have challenge in the recruitment issue, the dropout issue, and, possible missing doses, challenges in the steady state attainment. We also heard large susceptibility, and in vitro wise, some drug product has extremely low water solubility, so the real-time dissolution is impractical. So I feel and I think there are two aspects that we may explore, okay? One is under the current paradigm, which is the current steady state on the patient study design, what modeling and simulation approach could help, okay? For example, with regard to the dosing regimen, okay, if there are missing doses during the study, and more complicated if those missing studies takes place in the critical dosing time, okay? So how would we use this approach to leverage a study with this missing sampling times, or if a study has been conducted and some changes takes place later on, whether a modeling simulation can provide some information to bridge the result of the previous study to a new study, or to encountered changes, or from brother or sister applications. Okay, so the second aspect that I'm thinking is whether a modeling simulation approach would be able to support a BE decision PKB study design. Okay, we actually heard from our speakers some promising examples. Study design was a reduced subject population and a shortening of the study time. We also heard a single dose crossover study on healthy subjects and etc. Those are very interesting and promising, researches that we heard from today's presentation. We've already learned that modeling simulation has been used in supporting bioequivalence decisions in various of complex product evaluations. For example, recently we've been using, it has been used in inhalation drug product in some cases, the PD methacholine challenge B studies. And we also have some researches and examples of, using modeling simulation approach in COVID interrupted B study cases. However, none of such applications are as generalized as the long-acting injectables, which this whole class of drug product are facing to a fundamental challenge in the study design and the study conduct. So I do see from today's presentation and today's workshop, I do see a potential application of, the model integrated approach in this area of LAI. And I'm really looking forward to hear more from our technical expert on how to leverage this modeling and simulation approach to advance the science in this area. And more importantly and practically, how to translate the science into the real product application. So that's some, thoughts that I have collected from today's workshop. Thank you.
Thank you, Bing. I'm so glad to hear you are very confident and positive in terms of modeling can help to fill some of those gaps. So I'm going to turn to others who wants to chime in this topic. Dr. Liang, Zhao, can I turn it over to you? Then I will turn to others in the panel.
Yeah, sure. Thank you, Lucy. As Bing, also want to thank all the contributors today. You represent the best expert in the field. We have a common interest, as Bing mentioned, it's a very challenging area, but I do see that, given the information and knowledge collected in the new drug development, we should be able to take advantage of the knowledge we have gained to have more science-driven BE pathway for generic drug applications. And I do see from your presentations, there could be several areas, opportunity areas, including, for example, the study design. How, if we want to use model integrated approach, what would be the most efficient study design to have higher confidence to gain model credibility, and also kind of save the, cut the duration and reduce the sample size. The other opportunity area is how, how can we validate the model? Model validation certainly is based on the regulatory impact and the risk associated. And, there are some learnings as how presented from the new drug side, but in the generic drug side, depending on the purpose of use, we may need more stringent criteria for model validation. But to what extent should we consider the model being validated to drive critical decision-making in terms of bioequivalence establishment? That's also kind of a neat, expert's opinion for this issue. I do have mine, but I can share later. So the another opportunity area is that can we, as Andrew and others presented, can we also from industry presentations, can we have better PK metrics on top of Cmax AUC to, in combination with the study design, to better safeguard or control the type 1 error and safeguard the the quality of generic drug approvals. Those are the opportunity areas in my understanding, and I'm looking forward to gain all of your expert input in this very interesting but intriguing issue. Thank you very much.
Thank you, Dr. Zhao. I think that the comment I heard is more tied into how we can really implement from industry perspective. So now I would like to hear some thoughts from our industry colleagues in terms of if you in your mind, based on what you heard today, you think what might be the biggest challenge or the hurdle when you implement this model integrated approach in the development of generic LAI products. Any thoughts will be helpful.
So this is Ameya. So I think one of the biggest challenges, I think, from a generic manufacturer perspective is there a roadmap to approval? We can do a lot of development, we can do pre-NDA meetings, we can get input. And we have seen this in some of the other instances where there is development, but unfortunately there is no clear roadmap to getting an approval. So I understand currently we are in very nascent phases, but does the FDA have something in mind that they can put out which generic manufacturers can follow, which would take them to approval?
That's exactly the purpose of this workshop. We really want to work with all the stakeholders to develop the best practice. I think that it's not only FDA is working on this, it's like we also want to make everybody else get engaged in this, in developing this process, best practice together. So thank you for that. Anything else?
Yeah, this is Raja here. So basically, like what I'm thinking is it's a difficulty in establishing the model itself in the first place. We may have literature data, we may have other data, but actually developing that model for long-acting injectables, if we have something model already for the reference product, whether that can be acceptable. There is definitely a usefulness in terms of the study design, shortening the study design. We saw that today. So there is some room there, but how to actually, where do you draw the line that say it's validated? So the model has to be on its own, right? So it should not be product specific. The model has to be like a drug related, product release characteristic related, but not product specific. So that's the reason why we do that. So how do we say that model is okay at that point? So I think at some point, like you need to have a discussion with the FDA, come down and say, well, I have this data, I have this model. Can I just go forward with that? Like I think Ameya's already mentioned that somebody has to give some signal. If I spend a lot of money and time in this, investing in this, where am I going with this? That is my thinking.
Yes, thank you, Raja. I completely agree with you. The model validation is very important, and we will have in-depth discussion in our part 2 on this. So stay tuned. Yes, sorry, back to you, Dr. Chung.
Hi, Lucy. Thank you, Lucy. This is Chung speaking. I like what I saw today. I mean, that FDA is open to accept modeling for BE determination. That's the big difference from the past, where FDA used to be quite conservative in the sense that they don't really trust literature data. So it's good to see that perhaps FDA is more now open to accept literature data with respect to the PK modeling. And it's good to see that there are many potential use of modeling, and we have good examples presented today. However, what I didn't see today is how to validate the PK model, especially the population PK model, because it's great that you can do a lot of simulation, but if your model is not correct, all your simulated data would be incorrect too. So it is really important that we see how what way that FDA will accept validation of the population PK model, and too bad that we don't see any example today, because that it will be very useful to the industry to know what kind of expectation FDA has with respect to the validation. Thank you.
Thank you.
Lucy, can I make a clarification here, following in response to Dr. Yuqing Tang's comment? I think FDA, I just don't want to misinterpret the information. FDA do consider all the knowledge available in the field, including literature data, but we are not only accepting. It's not like saying we will, based on literature data, will make a decision making. But that's the distinction there. But I like your comment in terms of model validation. Certainly, for the long-acting injectable, we are discussing the best practice in terms of model validation. That's for sure. That's also the focus of the discussion here. Just want to make the clarification on that one.
Sorry, it's Mike speaking. So it's just probably, from Vitress, it's just probably reiterating and summarizing what's been said already. That there's been some very good presentations today that showed how models could be integrated into the clinical design, and there are a lot of advantages with that. And it comes back to what's the best way to build a model. Is it from literature, or is it from data that you would generate in specific studies for that purpose? And again, what is the best way to validate that? Because you are relying on the predictive power of that model to make some very, very key decisions. And again, the pieces are there, and it's just probably being able to flesh out a bit more how you can connect them together, I think would be helpful.
Any other? Oh, please help.
If I may, so I first of all, I wanted to thank the organizer. Actually have this nice section. I think it's really great presentations and also a great discussion here. So I totally agree with what has been said here. To me, conduct B study for non-acting injection has been very challenging. We have to get enough sample size in order to capture all the variabilities. We have to make sure the trial is not enough so that we have enough PK samples in order to get all the BE assessment, which will bring us another challenge. That is, usually if you have a long duration of trials, you have to face the fact that some patients might discontinue from the trial. So all the challenges are here. So using innovative tools such as modeling simulation does provide us alternatives in order to address this critical issue. So I definitely all for it. So having said that, I understand there's some technical challenges we also want to understand. We want to make sure that the model really captures the release characteristics from all the formulations, including the generic formulations and reference formulations. And also we want to make sure that, how to characterize the variability that goes to, how do we make sure the model validation, all the components are carefully evaluated before we wanted to, really put into assessment. But I think the tool is there. It's just how do we really refine the tool and make sure this tool is going to be suitable for our objective.
Thank you, Hao. Yes, please.
Can I make comment? I have 3 comments. I wanted to reiterate what Dr. Zhao indicated. I don't think complete in vivo waiver is on the table. So let's be clear. So there is a data that should be generated to support. This is my view that for LAI generic approvals, we will need to bring data for the reference and test. No question. Second point is that there was a comment about product versus the active moiety. The model is about the product. Let's be extremely clear about it. It's about the product. You cannot differentiate between the active moiety, and so it's an empirical approach really to going from a no path forward to a practical path forward. So we would need to characterize the pharmacokinetics of the test and the reference, but in a limited setting, abbreviated setting. Then you zoom or expand that experience if you were to do a full-fledged clinical trial. That's at least the notion that I presented. The second point. Third point, this is, I keep hearing time and again from the pharmaceutical companies, give me the prescription, I will follow that. I mean, I really discourage that kind of attitude to when we're talking about FDA is open to a new approach, and we are having this discussion. It's new in the generic world. It's not new in the NDA BLA world. We have Dr. Zhao here. He's probably done thousands of applications of such modeling in his division. So this approach is not new. We know how to validate models. We know how to design these studies, optimize them. But the attitude of saying that we are here to take notes and FDA tell us what is a prescription, we will not be collectively able to solve this problem, because imagine that if the generic world is waiting for this prescription for every product of exactly which model to use, exactly how to validate, exactly, then there is no innovation. So the companies also have a responsibility to contribute to this abbreviated path. It is innovative, although it's a generic copy product. The approach is innovative. So the generic companies will have to pitch into the innovation. They have to propose some methods to start with, and the agency would review them, and then, like they always do, give you good feedback. But I cannot see in the next 5-10 years that we'll have kind of a highly streamlined, guidance for every product in terms of how to exactly use the modeling. And I don't think it's a good idea actually to do that. So the generic company should have to pick up the weapon and stand opposed.
Thank you. I really appreciate that, Joga. Yes, Andy?
Yoga, I appreciate your comments. I think you made some really good points. So I think one of your points was that there's already a lot of information about how to validate models, that I think FDA has some guidelines about using models at least in new drugs, about how you can evaluate that the model is actually describing the system. And then I think Yu Cheng said that we hadn't shown any ideas about validation, and I tried to at least give 2 examples of how you might be able to validate a model in my presentation, where I talked about making sure that your model was identifiable prior to actually using it. So using techniques like Fisher information matrices to understand the uncertainty of your model before you run it in any situation. And the other was being able to simulate, using posterior predictive checks, the mean AUCs and Cmaxes that you actually see in your data. So that was at least 2 starting points for a validation that might be used for bioequivalence. But that said, I think that it's also maybe a fool's errand to talk about one model that is the model that's validated, and no other model could be used in the approach. So I think you're going to have a number of models potentially that could be validated in any situation, and then I think it's important to be able to use those models in concert in some way to make your final conclusions, because each model may have some specific properties that lead to different conclusions in some situations, and being able to understand those different conclusions, I think, is important. So it's something we see every day when we're looking at COVID predictions. You have some models that predict we're going to have a 4th wave. Some models don't predict it. and you can then see which models end up being correct. And I think the ones that do the best that have a weight of lots of different models that use the final, that make predictions of what's going on. Yes, that was, and I really like Yoga's point that we're not only the having a specific recipe is perhaps problematic, since every situation might be different, and we're going to have to be sort of scientific about it and make scientific reasoning for why you should use just this method in this situation.
So maybe I can also give a small response on behalf of a generic industry, just based on my interviews with them. I think Dr. Lee gave a very good introductory remarks. She said, she outlined all the difficulties the companies see: patients missing sampling, patients missing doses, difficulty recruiting, very long duration of studies, high expense of studies. All of those issues she summarized, and many presentations summarized. And then she also have an introductory remark. She summarized all the approaches we presented: having single dose and then going to another single dose, having parallel design, shorter complete dosing, washover, and then a single dose. So we presented several very different approaches. And I guess the point is that because we are Center for Research in Complex Generics, and the symposium is sponsored by the center, generic industry is the one operating on very small margins. So unlike large innovator companies, they don't have the power or funds to develop those models. Yet there is the goal is to compare their product to already established products that could be variable, and in populations that also could be very variable. So their model, and showing by equivalency in many cases, is harder than for innovator company to develop a brand new model. So the funds are not exactly there. The task is a little bit harder. And it's clear it's a big problem. That's what, Yoga, you mentioned. There is not enough approved drugs in those complex long-acting injectables, and it's a big economic burden to the United States healthcare. So I think I understand completely why some prescription would be of great value to the industry, to understand before they start going for those design of more innovative models and more innovative studies, to have a more clear understanding which way the agency is thinking. Out of all those proposed approaches, which one you think is the most viable? And then, and it could be a discussion. We could put opinion paper based on this symposium or whatnot, but having some sort of a guidance, I would say probably really could help generic industry. And okay, I'm an academician, so I would let the other people of the actual industry to comment on it. But that is my understanding of the problem. It's very little money, high competition, extreme expense of doing the studies the way they're prescribed right now, and huge risk of doing it wrong way and having to repeat it with the cost of completely dismissing this particular product development.
Dr. Schwendeman, I agree that there has to be some kind of information sharing that about the approach, the overall approach, like slides of what are the different steps to interact with the agency and gain concurrence and proceed with the plan. I completely agree with that, but not the product specific model. It's almost impossible for any one group to do it. And it's not that costly, by the way. I know it's this, we're both academicians. For generic company, they can afford it. It's not the modeling part, is not the cost part.
Yeah, I really want to thank Anna and Joga for sharing the thoughts from generic industry perspective. I think, as you guys may be aware, in the past few years, FDA actually has been in the driving force for the innovation. For like, we have been working really hard on model integrated evidence approach. I mean, we from our perspective, we work with internal, external experts. We think now is the time for us to actually turn those outcomes from those research projects actually back to the industry. So industry now can pick up the methodologies which worked in the past. That's kind of why we have this workshop. We think that it's about the time for us to really summarize what we discussed, what we learned, and now it's a time for how to move forward to the implementation aspect. So Miyoung, anything you want to add from FDA perspective?
Yeah.
Oh, okay, Mia. Okay, sorry. Go ahead, Mia.
Okay. Yeah, I have a very brief comment based on what Anna mentioned. So apparently we have more challenges because of the small size of the companies and resource issues. But at the same time, because we have resources from the new drug development and we have models that we can modify from. So I hope that we can leverage those available information, and we can discuss specifics about how to leverage those information from new drug sites for genetic drug development, especially in this area. Yeah, so Liang?
Yeah, I don't have any additionals. I see some comment from the chat, and I would say regarding dropout and missing samples, certainly including what being mentioned at the beginning, establishing steady state. I think models have a known advantage to take care of some of the data issues. Having said that, we are not encouraging, a lousy clinical study because the model can always be used to rescue study. But what I'm saying is that if that happens, I think models do have the known advantage capability to handle missing doses and patient dropout and missing samples. I think that's the advantage of having modeling approach. There are lots of relevant publications in the field, pharmacometrics already. The other, I think we've been focusing on the guiding principles a lot at the beginning. So rather than wasting everybody's time, I think we need to dive down probably to some of the technical details. The overarching message is that we don't have the best practice yet. We do want every stakeholder to have a fair shot in terms of integrating your thought opinion to make a fair program. We don't want to being super bureaucratic from the agency. Here you need a validate model this way, which entails like 500 subjects, and that's not the way you want to see in the future. And that's why Joga, Anna, and Andrew mentioned, we encourage, more like all stakeholder involvement in terms of rolling out the most science-driven and the most reasonable approach for the industry. So what I'm thinking regarding model validation is that there are two parts: the model center tendency, and the other is uncertainty. So I will say the guiding principle is that if the model can be validated to the extent that for the based on simulation, the type 1 error, especially for the agency, type 2 error for the power to pass a bioequivalence study for the industry, if those errors can be estimated with high confidence, we are talking about confidence on the uncertainty, right? If we have a high confidence on those uncertainties, then if the agency is fully convinced, then we are happy to agree with the model. Regarding with the study design, we are not, I think at the beginning, especially, we shouldn't be that rigid because there's no optimal practice established yet. Later on, after all of the effort we have been trying, then we see a very good, sharp approach and also reliable approach. Then certainly we can, download that in other platforms or venues. Okay, here's what we found. This being always working, always being efficient, giving industry benefit by using that particular design, by using that particular validation approach. Certainly we can do that in the future. I will stop here.
Yeah, thank you, Liang. I think we will discuss more in our part 2 in terms of the technical details. So now I want to switch the gear a little bit. I want to follow up with Dr. Hao Zhu's presentation on the 5.5b2 examples. So I hope you guys are convinced that modern simulation has been demonstrated as a powerful approach to support approval of non-acting injectable antipsychotics in the new drugs. So I would like to hear from you all, to what extent can we borrow notes into generic drug approval as well? So how can I start from you?
Oh, yes, definitely. So I think we have, like you said, we have tried to use modeling simulation to support the different strength or different product approval and different dosages. To me, I think this is it is the same philosophy that we can apply to support a generic product approval also. But I'm going to leave to our OGD colleagues to comment on that. But philosophically, I would say yes.
Thank you.
This is Raja here. Can I, just comment on that one? So,
yes,
in the
please.
505b2, we have a different formulation. That's the reason why people do that, and different, ingredients or different mechanisms of release, whatever. But they want to show bioequivalence. In this case, in the generics, we have in the long-acting injectables, we have Q1, Q2, Q3 mandate. So we have products that are lined up for this one. Then we are actually going into the BE study after that, right? So we have pilot study, like we are, there with the Q1, Q2, Q3, and then we are going into the pilot study. So the paradigm is slightly different. In this space, we are looking for really equivalence. So we're looking for a product that is maybe 10%, not more than 10% different, because then you don't pass the intervals. So you have to have a model that actually sensitive enough to detect that 10% difference between the formulations. In the long acting, you're talking about the release rates, right? Basically, release rates. The other one is the compound is the same. We are exactly the same. Ingredients are the same. Everything is the same, right? So the release rate is the only one that is uncertain that you're looking at. So it has to be within 10%, and not sampling in the areas where you detect the release rates in vivo is concerning. So, but at the same time, Liang, what we have is a pilot study or a small study, right? We all know that part we do. Industry is very happy with what we heard today in terms of doing a small study. Now, I didn't say a small study, but it could be a pilot study, or it could be a study of intermediate size. All we are lacking is the power. You don't have to do 500 subjects. Maybe you're doing 50 subjects, 80 subjects, something like that. and give you a certain power, right? So that data is certain. It's not like a loose data. It's not like lacking data. It doesn't have the power. So we can put the modeling in there and then say, well, if I regenerate, the data, more data, along the same lines with the same characteristics of the products, maybe I will have the power. So that is where I think is very powerful in terms of getting the bioequivalence determination or confirmation, I call it. So it is there, but you're confirming with the data, simulation data. That's where I think is useful. The second thing I thought is useful is for design. Yeah, a lot of designs came on, I agree, some of the designs we can work with, and that can be checked with the agency prior to the applications. Rob's point is well taken when he started, that we can come and do that. So those two areas I think are very useful, but do not take the pedal off from as a consumer point of view. If I have to take the drug tomorrow, am I going to take this drug knowing the data? That is my important thing to me. And not taking the samples or not avoiding the samples where the release rates are important to distinguish the product is not going to be working. So we need the data, but we may not have to have the data of 500 subjects or 200 subjects. We can generate the data with what we have. That's my point. Thank you.
I think we will discuss more in our part 2 in terms of how do we validate the model and what study will be considered adequate for validate the model and for your subsequent virtual simulation. I think I will hold on my comment to what you said just now, Raja. So I want to hear a little bit more from you guys in terms of like the experience we learned from those 552, and I really like what I heard just now, that the intended purpose is a little different. And for BE purpose, we really want to make sure the POP-PK model has the capability to differentiate the formulation differences. I think that's something very important for us to remember. So anything else? Miri, can I hear anything from you?
I had a question. So we've seen that kind of there's a low number of generics on the market, and say out of the 30-odd long-acting injectables, only 2 have got generic. And the approaches that we've seen have been based on there being a reference model available, a model for the reference product. But only about, I think it was between 8 to 10 of the 30 actually have a reference model available. Now, if say there has to be a model, if the regulatory bodies say there has to be a model as part of the initial drug submission, what incentive is there for the innovator to publish their model as part of the submission? Because if there's no reference model available, how can the generic then use that as a stepping stone for their application?
Anyone wants to take the question? Yes, please,
So
Miri.
can I ask Mr. Gajjar, why do you need a publication? Can I ask, why do you need the brand company to publish?
Yeah, I was going to talk about that, Yoga. Actually, most of the time, there is enough information in the NDA documents on population PK model that was submitted to the FDA. There's enough information in there so that we can recreate the model, even though it's not published, like Joga is mentioning.
Yeah.
We've been working with a few generic companies ourselves with long-acting injectables, and we've leveraged that information that is in the NDA redacted documents from the agency. And I don't think that the model that we take from those documents or from the literature is necessarily perfect. I mean, many times we see flaws in those models or things that could be improved. So we have to realize that. Sometimes there's no interoccasion variability. Sometimes the data that was used to build the model was so noisy that the residual variability is so high that it's not that useful for a bioequivalence trial where we try to be at plus or minus 20%. So I always see this published model or the model in the NDA as a starting point that we can improve. So that's why I think a pilot study, minimally, where you replicate the reference, is useful to validate externally or modify that initial model. Yeah.
I mean, look, Dr. Gajjar, I think that you will not be able to develop a generic without having a feel for the in vivo PK of the reference. You don't have to answer that. I will take it as a yes. You cannot develop a generic formulation, period, right? So you know the shape of the reference. Then the reason you would want to consider a model for that shape, how you do it, there are 17 different ways to get to that, but that information would be available to you somehow. You would use that to design your abbreviated trial. After the abbreviated trial results are available, then you use that data to build your model, gain confidence, model validation and all that, and then use that for the simulation. So I have a feeling that some of us are thinking modeling meaning you don't need to do in vivo study. I hope that is not, I hope I'm wrong.
Yeah, I echo you, Joga. Thank you to make that clarification on FDA stuff behalf as well. So you know, there's for the pilot study, I think even though currently we call it a pilot study, right? We do need a, you know, I think in the future, if you're using model integrated approach, the thinking could be what would be the data that need to be available to support your model credibility, you know, to make everyone, including the FDA, to trust the model you are using for simulation, right? Either individual-based projection or population simulation, as Andrew presented the model integrated approach using the model to simulate virtual trials. Either way is fine, but you need show us with you know, both test and the reference data within the same study that allow you to detect the formulation difference without getting worried about inter-study variability, variation. I think that's something you must consider at the current stage, at least. I think Andy want to chime in. You know, Andy, you know, give time to you.
Yeah, I just wanted to point out, I think it's, Yoga, I'm not sure if you were suggesting that you should build your model based on the actual BE study data that you might be doing. But I think that's dangerous if you're building your model on directly on the BE study data, because you'll be biasing your model towards that data, and your predictions, whatever predictions you're making will be biased towards just that data set. So I think you need to have a pre-specified model in these situations if you want to control type 1 error. But other than that, I totally agree with the idea that using some maybe pilot study to develop the model. And I don't mean to sound too.
I don't think we're disagreeing.
Okay, good.
Too much, but we are.
Too much.
We are slightly different. You will have to use the abbreviated B study as your pivotal data for the model. You could use other sources, like, for example, the Bayesian approach I was talking about. You can borrow information from other sources, but that study is critical, I would think, because there is no other information about the test product. I mean, So we need a reasonable estimate of the relative viability and the rate for the test product. That's my only source.
Oh, I agree with that. But then I think that for the test product, you need to have pre-specified components in your model that you will then estimate with the test product data.
No, why do I need to do that? I'm going to collect good data. I'm not going to collect, extremely sparse data that is not going to be informative to model building. I would collect, but in a fewer subjects, in a shorter study.
Yes, but
yeah.
I think that if you start changing your model and trying different models and doing tests between those models in a non-pre-specified way, then I think you hurt your chance of actually being able to control type 1 error.
Yeah, that's what we have been advising the applicants. Modeling, think about statistics before conducting study, you need a SAP, right? Statistical analysis plan. Yeah. So we also worried that, people could also abuse the model by doing lots of post hoc analysis until you pass the BE. That's the concern. So we have another terminology, so modeling analysis plan, model analysis plan ahead of time, that to say, given the data, I will do this, use this approach to detect the reference to test difference. Scientifically, that may not be ideal because many times there could be better models developed post hoc. But to be fair, for regulatory practice, we do think people need have a model analysis map, kind of a model analysis plan ahead of time in place before conducting the modeling.
Thank you, Liang. I think for the interest of time, I have the last question to get your thoughts on. So the next question is really touched upon the global harmonization. So if an applicant is conducting the BE studies based on the model integrated evidence approach framework, do you anticipate whether there will be any barrier to adopt the approach from other regulatory agencies? If so, what will be the solution? Particularly, I know some of your, from the industry perspective, you want to do one study that can satisfy the requirement from multiple regulatory agencies. So I want to hear your thoughts in terms of this. Yes, Miri.
This actually is a problem with every type of submission, is that normally for FDA, we only use the US reference product. For EU, we only use the EU reference product. So, I mean, it's much bigger than just the model-based, but regarding the model-based, the FDA is really in front of EMA and of Canada, for example, much more in front. And from what I've seen, my interactions, I've been very positive. Industry with us have been proposing things to the US FDA, and you've been very open-minded on everything, on validation, on every little steps, and given a lot of positive feedback. So I would think the other agencies will do the same, but they're behind, obviously, compared to you guys.
Thank you.
I don't know if I'm answering the question.
Yeah. It's always good to be positive. Yeah.
Yeah.
May
I make a comment on that, Lucy? So I agree with Murray that other agency will probably follow FDA, but I have to say that they're probably behind FDA in accepting modeling, because there's always the perception that modeling is complicated, difficult to understand, and more importantly, it could be highly dependent on the performer of the modeling. especially if you utilize simulation to simulate data, it is not impossible that the performer will simulate data that is to the best interest of the performer, increasing the chance of having positive outcome. It is possible to, especially with simulation, it is possible to do that. Unlike computer fitting, where you have strict statistical criteria in coming down to your optimal solution. For simulation, it could be very subject to personal bias, especially by the performer of the modeling, so of the simulation. So I think there's always a bit of distrust on simulation, on relying on simulation for BE determination. So to me, it is important that we come up with something that's easy to understand and less subject to the bias from the performer of the simulation. Thank you.
Thank you. Oh, go back to you, Mary.
Yeah, I just want to say something. I agree with what you said, Yu Chung, but one way that the FDA has circumvent that problem is that they redo all analyses, and they will do sensitivity analysis, and they will change certain things, so they don't accept just what has been submitted. They always verify it. Europe is doing the same thing on the new drug side when we submit population PK analyses. Health Canada has started doing that as well. So to me, it's obvious that on the generic side, they will do the same thing. They will redo the analysis. They will test it to make sure that it's okay and it's not biased.
Well, for
that,
I'm not sure about that's true for submission based on modeling. I mean, for simple BE studies, yes, it's true that the most regulatory agencies do try to duplicate the analysis to make sure that there was nothing wrong with the analysis. But in terms of modeling, like Health Canada, or well, maybe I shouldn't mention any regulatory agency, but some of them do not have the software or the expertise in doing it. They may do that in the future, but I don't think that's what they're doing now. It's different from the new drug side. New drug side has to have more tools, but for the generic division, I'm not sure they have that kind of tools.
Thank you, Raja, and then come to you, Anna.
Yeah, I think I agree, but this will be a dream, like for the drug industry to actually have one study that will register their product all over the world. To be honest, yeah, millions of dollars in savings. I agree that the FDA has put in a lot of effort over the last 4 or 5 years, and Liang knows like how much we put into this one in a workshop, developing the models, and like going back to Rob, like I was looking at one of his presentations from 2009 or something like that on paliperidone model. I was going, we are discussing paliperidone after 10 years. Okay, that's good. But I was thinking that, okay, so how do we actually make this work, for the FDA? Let's say I did a study in Europe with the European RLD, right? And then I, passed the test, whatever, like in European RLD, like we passed the PE and something like that. Can we use it for US? Do we have all the modeling tools now? We know how to model this one. We have a really good data with the BE. And then can we use it for US RLD? That's the one question where we can use modeling and reduce it. Or you do some bridging, so we may have like some pilot study or something like that in the US, but we don't have the full study. Can we actually extrapolate the data and bridge the data with the European data? Maybe that Canada or somebody will also honor this one if the FDA honors it. So we're thinking like, how do we use the modeling for bridging the data, not only just confirming the BE in the US, but also bridge the data if we have foreign RLD data, right? So why not? It's a model, right? So if we are really allowing the RLD data to confirm our model, maybe the European BE study, that probably is good as anything else, right? So that is something that we can think of to accept that. That is my thinking, Lucy, on whether the FDA can actually use foreign data to build the model and then use some test data for the US or virtually generate that data. I don't know. How do you feel? Thank you.
Okay, thank you. That's clearly a little bit more considerations than what I originally trying to ask. Actually, I lost track of time. So Anna, if it's okay, can we hold your comment to part 2? So we will take about like 10-minute break. So then we will resume our part 2 panel discussion. So for part 2, we will focus on more in-depth technical discussion so we can understand the most important elements when we implement this approach in the drug development and regulatory review. So our part 2 will start at 2:15. So please take a quick bio break and come back. We will go to the fun part. Thank you.